concentration. That's right. You gotta go post this and make fun of me, aren't you? It's like, yeah, she got it all wrong. Okay, so that's basically tonicity. It's important. It's just a couple points on the test, but still, it's important to know. So when you've got a higher solute concentration, it's hypertonic. And this is lower solute. And on the inverse, when it comes to osmosis, for the water going through the cell, your water is traveling through the membrane of the cell. The water is wanting to travel from the area of lowest water concentration in relation to the solute. So there's more solute in here, so the water wants to go in here to equilibrate the ratio of water with solute with what's outside. So your water is going to go from your higher water concentration to the higher solute concentration. Is that pretty clear to everyone? No? Anyone have questions about it? Okay. And so that's specified. And then there was another question like that on the other side, and it was the reverse. It had the 0.5 glucose solution, and it would have been this one. So it would have been in the hypotonic solution, and the water is entering the cell. And then your first question, the first page, is a hypertonic solution, and the water's leaving the cell.
So the first one,
Yep, this is the first one. And this is, what is the number on that one? I didn't do the numbers right. That's number 4 on that page, but I was copying it from multiple quizzes, and so there's several number 1s and 4s and stuff. So, and then I corrected it after I printed up 40 of them. That's the way it goes. All right, now on the second set, where we did the dialysis with the membrane, you'll get a question that looks very similar to this on the exam, and it's going to ask you for what the positive controls are. When you're reading that paragraph, I'm just going to draw a sample of the table. And basically you've got sulfate, chloride, starch, and protein. And then here's your dialysis solution, which is in the bag. And then it's asking you if it's a positive control. And then this over here is the beaker solution. And it's asking you if this here is a positive control. And of course the H2O was the negative. So with the dialysis solution, it gave you answers that this tested positive, this tested positive, this positive, and this was negative in the dialysis solution. And in the beaker solution, positive, positive, negative, positive. So when you want to know what your positive control was, read your paragraph and see where it started. So we pipetted 5 mils of solution containing sodium chloride and starch into the membrane tube. So we pipetted a solution that has sodium chloride and starch into your dialysis tube. Since they started here, these are your positive controls for that. And then in your beaker containing the water, you had sodium sulfate and protein. So in your beaker, you started with the sodium sulfate and the protein. So these are your positive controls for the beaker solution. Which means, since this one was not a positive control, but it tested positive, that it traveled from here to here. And the same for this one. It was a positive control for the beaker solution, but it wasn't present here to begin with, so it traveled through the membrane as well. Does that make sense? That's how I sort it out. It's easier if you just break it down like that. When you read that paragraph, start out, glance at your chart, see what you have, And then when you read your paragraph, go ahead and mark these ones where they started. And that where they started is where they're positive control. Okay? And then what reagent we used to test for protein, and what was the appearance? We used Bradford reagent, and it was bright blue. In fact, I think I